quería agradecer aquí al comité organizador que ha estado trabajando duro para que esto funcione y salga adelante a pesar de la situación y las condiciones en las que estamos. La idea es que esto es una jornada festiva, así que es científica. Lamentablemente este año otra vez no podemos acompañarlo de un pastoral de apego navideño, que era la idea original. Así que tenemos que hacer un poquito más sin esto, acompañando. Y nada, agradecer a todos que estéis aquí, dar paso a la organización y que os cuenten un poco cómo se va a desarrollar esta jornada, ¿de acuerdo? Y bueno, es una historia.
Okay, so I think like some people who don't speak in Spanish,
Yes.
so I will speak English as
Do that.
So, well, the rules are simple, and well, I think you know them already, but reminding them is always good. So we have about 17 speakers today, and all the names are in this very pretty box. So when we start, well, we will take the first name randomly out of the box, and well, the person we can hear. But then it will be each speaker will take, at the end of his presentation, a name and call the next person. Some of the people inside here are actually not presenting, so it will be a moment of public awkwardness and shame for the ones who are not coming. And yeah, but thank you for being here. Okay, so we are being generous with time because in the end we have not so many people. So we decided so you have 3 minutes to give your presentation, and it's going to be sharp. And so the speaker can see, if he wants, the time on the computer, so the audience can see it, but the speaker, there will be a timer going on. But anyway, there will be Elvira, our music girl. Oh, no, who's the music? There will be someone with the triangle. And so at the end of the 3 minutes, she will give a ting, and then we'll have 10 seconds to say a final word, like, I don't know, goodbye, and then maybe, someone will be ti, and then you must leave the stage. Okay, you can remove your mask if you're speaking. We decided on this with the, no?
So I forgot to say that.
Yeah, I mean, so the person who can speak while he or she is presenting can remove the mask. And what else? So the language is, you can decide to present in whatever language you want. I think there are a few people, at least 2, who are not speaking Spanish or Catalan. So, I don't know, decide in which language you want to present. And yeah, we are gonna record this, and it then will go on social media of the institute. So if you don't want to be recorded, or you don't want it to be online, then, well, you come to us, and, well, we will make sure that you don't appear. And, yeah, then there will be the voting, the most important part. So, okay, we prepared these analogical papers to give out, and we have pens for the people who didn't bring any pen, okay? So, we are giving 3 prizes. And so you just decide the presentation you like the most. So we decided to be the most interesting, so whatever is your criteria, the one you like the most, and you give 3 names, and the first person that you vote for will receive 3 points, the second 2, the third 1, and then, well, the person with more points, you know the master of this. Okay, and I think that's it. And that could be also a very official moment for giving the prizes of last year, with just 1 year of delay. So, last year, the winners were Julia, Eneko, and Chiska. Official paper as a stamp, and it doesn't say anything, but it gives you the power of going to us or to the administration and reclaim, ask for your prize. So, okay. Are they all here? Yeah, Julia and well, yeah, Julia. First we are here also receiving the same thing. Wait, I think that we received the wrong here. Okay.
Sonreír. ¿El premio del año pasado se puede decir o cuando empezamos?
So last year the prize was a voucher to buy goods at every week. They got different vouchers to buy whatever.
El del Mario fui.
Yeah, exactly. If they don't have one, something that you want, just ask them to do.
Okay.
Okay, so the people who don't like can also vote, of course, but it's only analogical, so you come down in the break and you get your paper. I mean, no, people online. Well, I don't know what to look for, so it's good. And, I mean, if you're in the institute, just come here to vote, and if you're not, you lose your chance to vote, I guess. Okay, and, but we need to know, well, okay, you can send email to, okay. And then the paper, I mean the voting cards, goes in here at the end. Okay. So, can you make a beginning thing? No.
Malo inocente, ¿no?
¿Qué?
Malo inocente, yo creo eso.
Ah, sale en el público, no, tienes razón, no, tienes toda la razón. Entonces vas. Para el siguiente. Yeah. Then please don't put it inside again. No.
Well, I prepared this in Catalan, so I'm going to switch one in English. So if it's a disaster, then bear with me.
No, it depends.
Yeah, I mean, is there anyone here that doesn't understand English? Probably. And there's people that don't understand Catalan. Okay, so my name is Marga. I started working here in 2019, and last year I was hired as a postdoctoral researcher at the Marine Ecology Group. So more specifically, I work at the Fish Ecology Lab, working with a fish called a turkey razorfish. This fish we studied in the field and also in the lab, and we measure its behavior. So other members of my group, they go to the sea and monitor the movement and other behavioral patterns, and we take individuals from the field, we bring them to the lab and also measure other behaviors. Basically, we measure anything you can imagine that a little fish do in their daily activities. So, for example, we measure at what time they wake up in the morning, what time they go to rest, how aggressive they are, and their daily activity patterns. My job is to, well, besides all of these behavioral measures, we also take a small sample of a film clip, and we take genetic information from this sample, and we can measure all of the genetic variability that this fish have. And my work specifically is to compare the differences in genetic variability with these differences in behavior that we observe on this species. And you would think, why is this important? So a member of our group, Martina, that she's not here today, she found that individuals have different activity levels, and those activity levels are consistent across time. So there are individuals that wake up earlier than others over time. In the same way, there are individuals that they are also more aggressive than others. And for example, during the fishing season, if you wake up earlier, you are going to be more vulnerable at being captured because you are going to be active more hours, so you will have more chances to encounter a phishing person. And also, if you think that individuals are bolder, so they are also going to go to the date more than individuals that they are more shy. So this results in differential. They are going to be more vulnerable at being captured. And so if these differences are being captured have a genetic basis, you think that there can be selective pressure against these individuals that wake up earlier, and they are also more vulnerable. So my work is to understand the effects of these genetics on being captured, and also how this can have a population effect at a population level of long term. And I do more things besides that, so if you want to talk to me, I'm here at the lab next to the copy room. So, thank you.
Don't escape your duty. Yeah, no, sorry.
Julia Castro.
Una cosa, if you want to introduce like your name and your group at the beginning, these didn't go in the 3 minutes that you needed. We started directly, but you can say now, after you say, you can start the presentation, but if you want to leave, I don't know, your very nicely, and start the time. Sí.
Okay. I'm Julia Castro. I started the presentation. I'm doing my PhD, and I'm in between two groups. I'm in the peace ecology group. I'm in the marine macrophile groups. And I started my PhD 1 year ago, and now I start. I'm going to talk to you about the influence of seagrass structure on the growth of juvenile fish. Nursery habitats are those habitats in where juvenile density, growth, and survival are on average greater than in other habitats. And habitats that promote a rapid growth of fish are considered good quality nursery habitats because they have sufficient food to make fish less vulnerable to size-selective mortality. Seagrass meadows are considered good quality nursery habitats, but we wonder if the mere presence of seagrass enough, or the structure of the meadow affects its quality as nursery. For that, the aim of our study is to study the relative growth of Diplodocus annularis. This is the annual sibling juveniles along 20 Posidonia oceanica meadows of the southern coast of Mallorca, which follow a gradient of structural characteristics such as canopy height, slope density, or percentage cover. We study Diplodocus annularis, which has a larval pelagic stage, and then it settles, and both the juvenile and adult states they are resident in the Posidonia meadows. We study the growth during the post-settlement period, and for that we use the otolith microstructure. Otoliths are calcium carbonate structures that fish have in their inner ears, allowing them to hear, sense vibrations, and balance. By policing them, we can see the daily depositions, because the otolith grows daily by making calcium depositions on the outer layer around the core. And by counting these daily layers, the distance between them, we can infer the relative growth of the fish. With these relative growth rates, we use them as a metric to evaluate the quality of the different meadows with different characteristics as nursery habitats for these fields, and yeah, that's it.
You're not prepared?
Okay, yes, hi. I'm Luke. I'm working in the Infibio group in the marine ecology department, I think. And I'm going to try to present in which organism I'm working and the more structure of this organism and why I'm working, like the relevance of it. So let's start simply with like a piece of hair that you can look at and you can see. And this is basically the smallest object that you can see by eye, more or less. And it's of the size of 0.1 millimeter. You have 1 centimeter, you have 1 millimeter, and you take 1 pen, and you can still see something. Then you have the organism that I'm supposed to work on, that I'm going to work on, which are called Chlamydomonas, or chlamys, as we like to call it. And they are 10 times smaller than that, like the radius of it. So it's basically the biggest organism that you cannot see. The motors are activated in such a way that actually I don't think we really understand right now, that makes the beating asymmetrical and that helps this organism to push forward or sometimes also to tumble around. If you look closer at one flagella, you have this micrograph here. So it's basically a big churros. I don't know if you see from here, you have the lines here that remind of a churros. So this is the base, that part, and this is the tip of the flagella. And if you look even closer, you see that there are something we call microtubules, and there are 9 of it around. You can see here, and it's like doublets of microtubules. So it's like a really complex structure, actually. And between these, you have something that we call dynein arms, that are molecular motors that use chemicals to actually make the tubules slide between each other, and the sliding, because of the constraint in the structure inside, for example, some links that tie the tubules together, will make the flagella bend like this. And this bending produces this pattern that you see here of like simply a bending. No? And this is what produces the motion, but there are many things that we don't understand yet, and that's what I'm working on.
Good morning. My name is Tintuni. I'm working on the TMO department, basically in physical oceanography. Marta, I know I'm putting here.
A moment, please. And we have a question in the chat.
Okay.
Maybe, entertain.
Yeah. I'm half French and Irish. My father is Irish.
Okay, so Madeleine is saying that she cannot hear us well.
Maybe
So,
if you present from here, the mic is here, so you can try.
yeah,
Can I
Madeleine,
move it like this? Yes, so
if someone from the audience can tell us if there is any improvement. I don't know.
Can you hear us better? Okay, sorry if I touch my hair a lot because I'm going to the hairdresser next week. It's like really annoying.
Okay.
I'm sorry. Okay. Okay.
in seconds.
Yeah, the topic I'm working on is the extreme sea levels in the Mediterranean Sea that are caused by storm surges and wind waves. So a wave, everyone visualizes what is a wave, right? A storm surge, maybe not. So storm surge is basically an abnormal rise of the sea level at coast above the mean sea level under generally a storm. It's caused by different processes. So the first one would be atmospheric surge. So it's the weight of the atmosphere above water. If there's a high pressure, it pushes water down. If there's a low pressure, the contrary happens during storms generally. Then there's the wind. So the wind kind of pushes water toward coast. And finally, the effect of waves closing to the shore that are breaking. There's an accumulation of water, and this might be threatening for coastal populations or anthropogenic structures. So, when I talk about storm surges, it might not ring a bell, but we all know about hurricanes happening in the Caribbean, in the United States. For instance, the Katrina hurricane that caused a lot of damage because of high winds and high waves breaking on the shore. And some would say no such storm exists in the Mediterranean Sea. Or like if you surf in the Mediterranean Sea, they will call you a surfer loser because they think there are no waves. But it is true that the Mediterranean Sea is a semi-enclosed basin, and the wave generation is kind of limited because the distances, which is called the fetch, over which the wind might blow, is really small. But we all remember, for the ones who were here, the Gloria storm that happened last year, that was really damaging in Morocco, in the coast of Catalan coast. And this was taken, you can't see much, but it was taken into the calendar. So we have a good day for surfing. So what I'm working on is the generation of new handcast, which is basically the reproduction of the sea states every hour for 70 years. Okay, and what's the point? When you go fishing, surfing, or for field experiments, you need to know what's the weather conditions. Otherwise, this might happen. It might be in a storm with big waves or whatever. So similarly, when you want to design a harbor, you want to study the effect, interaction between wave and Macedonia, for instance, you need to know the wave climate and the occurrence of some surges, extreme sea levels at each point, right? So, for instance, when you design a dike, you take a harbor, the client is going to say, I want you to design a dike that will be able to protect my infrastructures for 100 years. Okay, so the tool we can use is a computation of return levels, which is basically what is the highest waves that might happen, the highest storm surge level within 10 years, 20 years, 100 years. So from probability. Thank you very much.
Jorge Ramos Alcantara.
Well, hello, I'm Jorge Ramos. I'm in the sea level and climate research team, and I have to wait until the presentation.
So apparently the people on Zoom didn't see the message. They were, yeah. So give me one.
Okay, so I'm going to give you an overview of the work we are doing, the framework of the Venom project. The main objective is to develop a low-cost instrumentation to implement a sea level measurement network. And we also want to develop or to perform a sea level and deep sea level characterization in the Balearic Island and the Mediterranean region in general at different time scales. Here we have a picture of one of our instruments. The DJs are based on an Arduino environment, and they have a acoustic sensor and a solar panel to power the circuit battery. Currently we have 21 instruments already installed all around the Balearic Island, 12 in Mallorca, 4 in Menorca, 2 in Cabrera, and 3 in Ibiza. Data is averaged and stored every 60 seconds in a micro SD card, and a 10 minutes average is sent to the transmitter module via GPRS2G and displayed using the Grafana multi-platform tool, whose interface can be seen here. And quality control has also been developed. There is an example here for the Fornes tape gauge TV. In blue we can see the correct measurement. In yellow the doubtful one, and in red the wrong ones. The high density of our network allows us to develop a characterization of the sea level in with a never seen before temporal and spatial resolution in the Mediterranean region. And among the phenomena we want to study are, for example, the risadas or meteor tsunamis, which are long and destructive waves induced by local atmospheric conditions, and that can cause sea level oscillation of even several meters in less than 1 hour. They are especially destructive in the port of Ciudadela, as we can see here, and we have also plotted one of these events captured by one of our instruments. Other phenomena we want to study are, for example, the platform wave or the extreme events, and we can also identify the main sea level variability modes. In conclusion, the tape gauges are a fundamental source of information of sea level, and we can, although they are point-wise measurements, we can reconstruct the sea level all along the coast exclusively from tape gauges. We are actually currently working on that. We are doing reconstruction in the Western Mediterranean, that's based on an optimal interpolation. We have found that the reconstruction reproduces significantly better the coastal sea level than the coastal altimetry. We hope to publish this soon, and that's all. Thank you very much for your attention. Daniel Rodríguez Carri.
Hello everyone, I work in the team of group as well, physical oceanography, and I'm doing my thesis with Nanda and Sido. So our work is mainly focused on the vertical velocities in the upper ocean. I will not present kind of the results or what we are doing, because it will be like too detailed or too much technical stuff. But I will instead present like our motivation to study this. So the vertical velocities in the upper ocean are really important because they act as they produce an exchange of water property between the water surface and the ocean interior. This exchange is really important because there is an interaction and an exchange of water properties like temperature and salinity, and between the surface and the ocean interior. This produces, thanks to this, the ocean absorbs heat, carbon, oxygen, and pollutants. The heat and carbon absorption helps to regulate the Earth's climate, which is really important, and also the absorption of rich waters in oxygen to deeper layers of ecosystems to the. Also, there is an upwelling of water rich in nutrients, which allow rich ecosystems on the surface as well. Here you can see a subduction, because of an ideal subduction that we would like to study more. And you can see here the phytoplankton layer, and the phytoplankton wouldn't live on this depth because there is no light. So having phytoplankton there means that a current is pushing the bottom. But they represent a challenge because they are 3 to 4 orders of magnitude smaller than the horizontal current. So as you can get an idea, we usually measure the horizontal currents in centimeters and meters per second, but instead the vertical currents we measure them in meters per day because they are much smaller. So this is a problem because these subduction currents are not as straightforward as can be seen here. It's more like as they are going down, they are being affected by the horizontal current. So the pathways, the three-dimensional pathways are really complex and are difficult to understand. What dynamics shape them? What are the properties from the surface that are being subducted to the bottom? Because also we have water subduction that goes from the surface to 40 meters, that then it goes upwards again. But so it's not always that we have this subduction from the surface to down 200 meters. So we are trying to resolve this problem, and also the scales, which are the time scales, the space scales, because there is a large variability. This subduction occurs in really small places and with a large intensity. So, yeah, perdón.
Hello everyone, I'm Pau. I'm doing my PhD in Este Ecological with an atravesé. Well, I'm going to present in Spanish because I think I need to explain all the results in 3 minutes. But then in the coffee break, if any English speaker is interested in the topic, I can explain it more slowly, more than 3 minutes. So, okay. Entonces, en este trabajo, nuestro objetivo principal era relacionar tendencias de fenología debidas a datos en Cataluña en más de 100 sitios de estudio. Entonces, lo primero que hicimos fue analizar las tendencias poblacionales de las especies y vimos que más del 50% de las especies redujeron su abundancia en estas últimas décadas y por tanto estarían en declive. Y posteriormente analizamos la fenología de las especies, cómo ha cambiado, si se ha avanzado su ciclo vital durante estos años, y contrariamente de lo que podríamos haber pensado, de que en un contexto de calentamiento global se habría avanzado la fenología de muchas especies, vimos que no, que la mayoría de las especies no avanzaron su fenología. Y posteriormente lo que hicimos fue analizar las tendencias climáticas y vimos que en la región, a pesar de que la temperatura aumentó en más de medio grado, al analizar las tendencias climáticas para periodos concretos, para meses concretos, vimos que en determinados meses la temperatura no aumentó, sino que se redujo. En meses como febrero, marzo y mayo, la temperatura se redujo durante estos 26 años, y justamente estos meses son clave en el periodo crítico, forman parte del periodo crítico de estas especies. ¿Qué quiere decir eso? Que coinciden justamente en el desarrollo de las fases inmaduras de las mariposas, y justamente son el momento donde la temperatura tiene un efecto más importante en la fenología de las especies, ¿no? Y esto en parte podría explicar la ausencia de este patrón que sí que se ha observado en otras regiones, ¿no? Donde la fenología de las especies ha avanzado de forma muy importante. Entonces, luego lo que quisimos hacer es relacionar estas tendencias fenológicas con la abundancia, con los cambios en la abundancia de las especies y analizamos pues, si estas tendencias fenológicas tenían un efecto directo en las tendencias de las especies. Vimos que no, que esto no tenía un efecto significativo y lo que hicimos es usar otra variable que es la plasticidad fenológica, que es cómo las especies responden a este incremento en la temperatura. Y vimos que las especies más plásticas son las especies que han reducido menos su abundancia o que tienen tendencias más positivas. ¿Qué quiere decir eso? Que las especies que se adaptan mejor al clima, que cambian más su fenología, adaptan más su fenología a la variabilidad climática interanual, son a las que le ha ido mejor. Son las que han tenido tendencias más positivas y por tanto, pues este sería un poco el resultado más novedoso del trabajo, de cómo esta plasticidad pues, puede ser muy importante en el contexto de la aprendizaje.
So we're doing the break in other two presentations. So by now we have seven speakers. So the two presentations will be halfway, and we will have a 15 minutes.
Okay, so I am Eneko. I work as a postdoctoral researcher at the group of the Psychology group, working with Trippi and also Marga, Arancha, Institute. And I also prepare my presentation in Spanish, so sorry for that. I will be also happy to chat with anyone outside. And I hope that with this nice sketch you will be able to follow more or less what I'm saying. Porque en nuestro grupo trabajamos en ecología de peces, ¿no? Y como todos sabemos, los peces son un componente fundamental de los ecosistemas marinos, pero es una fuente de recursos muy importante para el ser humano. Los recursos que nos interesa gestionar de una manera sostenible. Y para hacer esta gestión necesitamos información sobre la biología de las especies y a obtener esta información, que sea aplicable para la gestión, es a lo que nos dedicamos. La tecnología principal que utilizamos para hacer estos estudios es la telemetría acústica, que se basa en unos dispositivos electrónicos que son unos sensores que emiten señales acústicas y van implantados dentro de los animales que estudiamos. Y luego una serie de receptores, que son unos hidrófonos en forma de cilindro, que están programados para detectar estas señales. Y los ponemos por el área de estudio en el fondo y van obteniendo la información de los peces marcados. Es una tecnología que ya lleva años, pero que en la última década, gracias a todos los avances tecnológicos que ha habido, se ha hecho una tecnología muy flexible, que nos permite hacer estudios de muy alta resolución, porque nos permite obtener trayectorias como si fueran de GPS en un gran número de individuos, y por otro lado, con no tanto detalle, pero sí también nos permite hacer estudios a gran escala, apartando las escalas temporales y espaciales enormes. Por ejemplo, ahora ya hay transmisores que la batería llega a durar 10 años, lo que nos permite hacer estudios sobre individuos sobre gran parte de su ciclo de vida. Con estos datos, bueno, esto lo hemos aplicado a diferentes especies. Por ejemplo, hacemos estudios de alta resolución en el caso del rao, es una especie muy carismática aquí en Baleares, y la otra tecnología más a largo plazo la vamos a empezar a aplicar este año con veros, con especies ya más longevas de tanta. ¿Qué clase de datos podemos obtener con esa tecnología? Bueno, aquí es como unos ejemplos, hay muchos más, pero me dio un poco pereza dibujarlos. Entonces, bueno, nos permite medir las interacciones que pasan entre los individuos en el mar, lo que nos permite caracterizar la estructura social de las poblaciones. Por ejemplo, esto con el rao lo que hemos visto es que se estructuran, tienen una estructura de tipo harén, en la que hay un macho que interacciona con muchas hembras, pero que luego esta harén no interacciona con otros harenes que están ahí alrededor. También nos permite obtener información muy importante sobre el uso del espacio de los hábitats. Nos permite saber dónde establecen sus territorios, por dónde se mueven, si hay zonas donde se agreguen o no, que luego pueden ser zonas prioritarias, por ejemplo, para la conservación. Y por último también, otro aspecto que nos permite estudiar es cuál es la variabilidad natural que hay en el comportamiento en el mar entre los individuos. Pues ya sabéis que todos los individuos no se comportan de la misma manera, hay individuos más agresivos, más exploradores, que se mueven más, se mueven menos, y esto luego tiene repercusión también sobre su capturabilidad, por ejemplo. Y de hecho todos estos factores también interaccionan entre ellos. Hemos visto, por ejemplo, que su comportamiento va, en el caso de RAO, está muy determinado por el harem al que pertenecen. Y bueno, y todo esto lo que nos permite es al final conseguir información que nos permita conocer mejor las especies para poder gestionarlas de una manera sostenible. Next speaker is Diego. Bueno.
Okay. Good morning, everybody. I'm Diego Rea, I'm a technician. I work with Iris and Leisha. I want to show a little work about greenhouse gases. Okay? I call it a spatial temporal variation of the emission in the coastal area of Mallorca in Western Mediterranean. So I want to separate in three very important steps. The first is what is the problem, no? You know that the problem is like we produce greenhouse gases and we need to measure it and estimate what is the flux between the ecosystem, the sea and the terrestrial. So we know that it is three principal greenhouse gases, that's CO2, methane and nitrous oxide. So if you see the figure 1, you see in the last century the methane increased very fast, you know? So the problem is we need to measure what is the estimation of the flux between the ocean and the sea and the atmospheric methane. So we choose 3 points very near the coast with 3 different anthropogenic pressure to understand what is the production of the fluxes from methane on the coast of Mallorca and Cabrera. The first point what we choose was Buenos Palma with high anthropogenic pressure. The second point was Captain Salines with medium anthropogenic pressure, and third point Santa Maria Bay in Natural Park of Cabrera. So what we're doing to estimate the fluxes of methane on the coast, we take samples and take data sensors with, for example, temperature and salinity, and for 3 years. So we principal resort are in the 3 points on the coast. We have like the same behavior on the 3 for temperature and salinity. We have in water geochemistry, we have clear seasonal pattern in alkalinity, for example, and pH. But we have a different comportamental of dog that is dissolved organic carbon. We related, maybe related, we don't have proof, but related with the holy seasons in the summer, with the tourists. We need to try to understand for what. And what we find in the Parthenon methane, the concentration. So we have 3 different points. Bay of Palma, we have, and Santa Maria Bay and Captain Salines present 2 peaks, one in summer, and the second, a lower peak in winter. We related this with the production on the sun, of course, because the coastal source produces a lot of methane in the sea floor and the water column. And we estimate what is the difference between the years, because we made a sampling for 3 years. So, for example, here in the 2019, we found this estimation, and we have a source of methane on the coast of Mallorca. And thank you.
Yeah. Oh, yeah. So
15 minutes break or
thank you.
Jorge Rodriguez.
So sorry, an introduction, a change. Now we decided to put the names so that, I don't know, if nobody, maybe we don't know each other. So if you want to write your name when you come, so that the people who want to vote you know. I mean, this should be the order. If someone is missing, or the order is not right, tell us. But I think it's right.
You said that we can speak any
Yes,
language that
so-
we want.
Right.
No.
To you, no. Then you face the consequences.
I don't know how to solve it. No,
Sorry,
yes.
technical problem. Password will be as computer.
So I work in the Fisher College group with Pep, and well, I will tell you one of the works that I have been doing recently. So well, I am Jorge. Then this work we are doing with Victor Aguiluz from IFIS, with Ana Sequeira from the University of Western Australia, and with many co-authors that were contributing with data. Because we have a big data set of animal movement in the oceans, and then we want to extract corridors. So how many of you like cooking in Christmas? So please raise your hands. Oh yeah, I see many of you. So then, as you like cooking, and I also like cooking, so I want to give you a recipe. So a recipe because I see that you guys are doing very cool things. You are doing experiments in the lab. You go to the field and you do very cool experiments. And then I'm the boring guy that stays in the office with the laptop and doing some analysis. So then I will give you a recipe of the kind of analysis that we do. So imagine that we have a trajectory of an animal in the ocean. This is the black curve, okay? And this is the real trajectory of the animal. We are never going to know the real trajectory of the animal, so we are going to have some location estimates from our sensor. So these are the orange stars. So we have some estimates. And then we can more or less know how the trajectory is. And then we divide the space in grid cells. Okay? So the idea is I want to know the direction of the animals when they arrive to a specific grid cell. And how do I do this? So basically, if I look from here to here, maybe just the animal went here for a specific reason, but I prefer to look at the long term. So I prefer to wait a little bit to see really where the animal is going and forgetting about the noise that we have in lower temporal scales. So then I look and where the animal is after this capital T, this time, and then I build a vector, okay? I build a vector, and then this vector has an angle, and this tells me the direction. So to now, this is very nice. So, okay, we have one animal in one place. What happens if we have several animals? Then what we do is we have many vectors. Each color corresponds to a different animal, and then we make the average of these vectors. We sum all the vectors, we make the average, and then this gives us a direction. How do we build a corridor? So for every grid cell that the animals are visiting, we have a direction, and then we are joining cells that have similar direction. This cell and this cell have similar direction, we put them together. And then we put all the cells together, we have a corridor. What do we want to do here? So we want these directions, and we want to explain how the animals are moving in the ocean, why they are moving the way that they are doing. So we are going to look at correlations with the direction of magnetic fields and with oceanic currents. So this is basically our work, and well, if anyone wants to talk about it with me, I'm happy to do it. Colin, Maria?
Okay, so hello everyone. I'm Pauline. I've been on an internship here for 6 months in the team of group, also under the supervision of Marta. And today, Today I will present my work, which is on the topic of extreme sea levels analysis, predictive like decadal forecast along the European coast. So when we think about storm surge, Tim has already explained it. We have a lot of problems with hazards to coastal infrastructures. So we would like to be able to predict these extremes, or at least know how they're evolved in the context of climate change, so in a decadal timescale. The problem is that we can't really do that with tide gauge data, because extremes are caused by storms, which are rare and strong, so they can even destroy the device, and so you have no data. And it's impossible to predict with tide gauge data only. So what we can do to be able to do what we want to do is to use simulations. So to have more data to make statistics, you use simulation, so hydrodynamic or statistical models to create other possible climate realizations, to have like other possible things that could have happened. So what I'm doing in this internship is that I adapted a statistical model developed by Michael Tadesse and Thomas Val in 2021, which aims at retrieving the relationship between wind and pressure, so the outputs of our climate models, and storm surge. So Tim has already explained that, but basically it's the abnormal sea level rise during the storm above mean sea level, and this is our indicator for extremes. So we have observation from the tide gauge, and our predictors, which are the outputs of the model. We train with machine learning, and then we take the parameters and reconstruct to have more data, and we do that all the time. And for those of you who are not really, who don't really know machine learning, I've put a little cartoon that explains very well what is happening there. Basically, you just tell your computer, well, the answers are not good enough, try again, and do it again until that's good. And you don't know what's happening in there, just after it works, and you're like, okay. And so after that, the goal, when you have the reconstructed storm surge, is to carry out extreme analysis, so plotting the distribution of the extremes on time to see if the final goal of that is to see if the climate, like the system described by climate models, by these climate models I'm working on, is predictable and to which time extent. So thank you everybody.
And now I'm going to explain a little bit what I'm doing during my thesis. Okay, so let's start. When ecology meets geometry, involves the term of self-organization in nature. By definition, the self-organization is a process where in which the pattern at a global scale emerge from interaction between lower level agents. So in the ecosystem, we also find this spatial self-organization. And if we want to approach to it, we may ask, why do they require this disorder and structure? And by answering to this question, we will be improving our mechanistic knowledge of ecosystem, and we will be facing the possibility of catastrophic presence to collapse. So during the last century, the fairy circle phenomenon was shortly described in the Navi Desert, and there are 4 theories under discussion, and I will use them to compare with our insights into the fairy rings of Oceanic ceramic matter. So the main question here is, what why is this ring-shaped pattern appearing? So in IMEDEA, they found that the presence of gas into the soil and inside the circles could be killing the plant. Similarly, we thought that the presence of sulfites into the sediment could be causing the emptiness of the branches. We confirmed certain concentration of hydrogen sulfide into the sediment, so the plant could be killed. But why is this killing following the pattern? We cope with it, developing a mathematical model with an equation for the vegetation and another for the sulfide dynamic. And then there was the interaction for producing the pattern. And for the vegetation, it matches so well with what we found on the sea. But the pattern of sulfides was less clear, so this discussion is still, so the theory is still under discussion. Then we went to a self-organizing theory within IMEDEA. They found that the fairy circles could be emerging from competitive interaction for water. So under the sea, we detected a specific arrangement of shoots inside our rings. We found that the oldest and the more vulnerable shoots were always in the inner edges of the ring. So for sure, if they have to die for competition, self-shading, or any other interaction, they will be from the inner to outward, then reproducing the pattern. So these are the main words of my thesis, but we are still looking for more agents that could be producing this pattern, because we don't see these same patterns in the most of the middle of Poseidon. So thank you very much.
So someone is doing the triangle. Okay, so I'm IMEDEA and I'm starting my PhD. Well, starting, but feels like that I'm starting. I started my PhD with Iran, and I'm also in the TBO group. And the work I'm presenting, it's not my thesis, but it's a collaboration we have with the ICM in Barcelona, and we have been working on it in the last months. So, okay, so the question that I wanted to address was like, how do microzooplankton sense its prey? So what you see here on the top right are two predators of the planktonic food web, Gyrodinium and Oxyris, and they're both herbivores. So they feed on microalgae, who are usually also swimming around in the ocean water. So one of their favorite food is this ugly thing over there, Rhodomonas salina, and which is a macroalgae. But the question is, like in this world in which nobody has eyes, at least in the way we think of, how do the predator know where his prey is? So how does it know in which direction it should swim to find food? So one of the theory is that the prey is producing some chemical compound, actually a series of chemical compounds, and the predator, the easiest thing would be that the predator just is sensible to those chemical compounds, so it can sense it, and then it can follow the chemical gradients in the water and get to the food. So the experiment they are doing at IPM is that, it's a typical chemotaxis experiment. So they offer to the predators two capillars, a choice between a capillar with a chemical compound, which is released by its prey. So the predator should think that there is food there, and then a capillar with normal seawater, and they put it under a microscope and see what's happening, and they film it. And then what we do here is that we take all this video material, and so all those tiny dots that you see in the photo are actually, each one of them is a cell, it's a predator. And what we do is that we do some video tracking of the experiment performed at the ICM, and we can reconstruct all the trajectories that the cells are following. So we can follow each singular cell, at least we would like, we will be able to follow each singular cell and reconstruct the paths that the cells are swimming at, and that are following while swimming. So we can produce this very psychedelic image. So what we see here, for example, is all the trajectories followed by all the cells during one experiment of 10 minutes, and you don't see anything. But actually, we are able to follow each individual trajectory. So by following each individual trajectory, well, you can know, having a big statistical sample, in which direction they like to swim. Do they go towards the chemotraktion capillary? Do they speed up when they try to go there? Do they change their swimming behavior? And, well, we still didn't answer to all these questions, but we are on it, and it seems it seems that we start to see something. And spoiler alert, it seems that they are. So, Rothman. I thought it was Rothman.
With T, yeah.
Okay.
Yeah, it's a strange name.
Hello, good morning everyone. I'm Rothman Criolla. I just arrived here in the island from June. I'm doctor in hydrologist and geological engineer, and I have a Margarita Comas postdoctoral contract from the Balearic Island, and I am with Alejandro Filahan and in collaboration with Victor Vilar. So my research line focus on the shallow groundwater, shallow geothermal energy is installed in the coast areas and how it may affect the groundwater discharge and the saltwater intrusion inland. So this kind of green energy is currently increased the installation of these systems, and mainly because of the cost of the regular energy is so high that it works to invest in it, and second, because the big jump of the technology installed that allow to optimize the exploitation. However, there are few studies addressing how this shallow geothermal energy affects the environment, especially in the coastal areas. So, as you know, temperature and salinity are key parameters for coastal ecosystems and for the kinetic equilibrium of rock and water. That is very, very relevant in areas like the Balearic Island. So I will address this research by two ways. The first one is by field campaigns that I have not started yet, and the second through numerical models having into account the mass and heat transport in groundwater. And to achieve that, I am currently using the finite element model called FIFLOW that is developed by DHI. And this, and I show you here the conceptual model of how these mechanisms interact among them. And here is direct from the oven a preliminary results of how these mechanisms play each other. So you can see here the fresh water, the groundwater, towards the coastal zone, while the seawater infusion downwards to the inland. Yeah. So and at the same time, you can see the thermal plume that goes to the sea, while you exchange energy with the subsurface. And yeah, that's what I am doing here. I'm working on it. And if you have any questions, I am on the first floor close to the HANO office. Thank you.
Dario.
Sorry, can you confirm me that there are just 2 speakers left?
One.
Are you okay? Yeah. Okay, so good.
Hi, I'm Alexandra, I'm part of the DRS group, supervised by Victoria Larasa, that is also here in the second floor. I'm in the first one, and yeah, I think I can start wherever you want. Okay, so to explain a little the motivation of our work, there is like a general complaining about this climate change and how is it related to CO2, and in this case I put this slide just to show you the IPCC. They reported different scenarios to address like the average, the average warming up of the temperature in the planet, and to keep it less than 2 degrees, as you see from here, maybe not so clearly. We have just these 2 pathways, which are the blue ones, and the blue ones after 2050, it is expected to reach negative CO2 emissions. So to address this task, one very promising approach can be to inject CO2 into the deep soil, no? Either directly taking it from the industrial activity or taking it directly from the air. So this is the main task of the CO2 long-term periodic injection experiment, POLPI, that will take place in Monterrey, which is in Switzerland, not in Geneva. But I've provided in the center of the slide on the top the photo of the excavation front nearly in the place in which it will take place. This experiment basically is in a shale. The shale, for the ones of you that are not so familiar with this, as a behavior, which is a cap rock, like this layer. What is the problem? The problem is that when we inject CO2, this creates a plume, which is this blue stuff, that since the density of the plume is less usually than the in situ grind, these tend to go upward, then reaches the cap rock. So to ensure that this CO2 that we inject does not return up, we need to ensure that the cap rock is doing its job. So it keeps its sealing capacity for tens and millions of years. But the design side of this project, what we do is modeling, basically, and we also work with computers, in this case is a finite element code, classical finite element code. We test like cyclic, in this case, injection in the injection part, to be able to describe also a bit of a physics, also the shapes as a particular thing that we call bedding, so a particular direction in which you have like preferential flow path for the CO2, and you can see from here a bit like the pore pressure after 2 years we simulated it, and the pore pressure distributed more on bedding today. I don't know if I have any time left.
You're perfectly on the 3 minutes.
Maybe. Yeah, that is.
The pleasure of last but not least,
I'm Mary Kane. I am a postdoc researcher with tuvo in the Bio group. So I work with the amazing IMEDEA and Luke who put this together. So my question, primary reason for coming here is that algal blooms are becoming a more consistent problem over the last few decades. And particularly in the Mediterranean, we get a lot of these blooms where the water is green. This is from a bloom. And we get lots of this green water, which not necessarily toxic always, but you don't want to go swimming in green water. I certainly don't, and I do it all the time, so I'm nuts. But the question we have is, can parasitoids actually be used to control dinoflagellates? So like, can a parasite be used to control these populations in a little bit more artificial method? So like how, if you're familiar with some farming, maybe in the States, how ladybugs are used to control aphids like a natural predator. And so there are 3 parts to the project I'm working on when I come here. The first one is natural sampling, which involves going to Cala Santani, looking at the phytoplankton population using this instrument, which is called the flow cam, and then getting all of these lovely pretty pictures of different phytoplankton, and hopefully using these images to find symbols of infection, and particularly looking for parasitoid that we're looking for. Also in the future, what I'll be working on is laboratory experiments with IMEDEA to understand the individual interaction, so at the micro scale, what's going on between individual dinoflagellates versus individual parasitoids, and then also combining these larger scale natural samplings with the individual behaviors to try and get a better numerically accurate model, complete with validations using experimental methods to determine can we actually exploit parasitoids as a way to control algal blooms in the Mediterranean.
We are actually we define the schedule, so I don't know.
And once you're done, you can give it to us.
And well, thank you for coming.
So as you can see, there is no corruption. I'm not from the organizing team.
He's my friend. Close friend.
Yeah. Bueno.
Thank you for
Thank you.
Thank you. So, for example, Mary doesn't receive the emails.
Yeah.